Hello, students. This is Fazan Mirza. We are discussing the process of meiosis right now. Let's start with the discussion of genes. Genes are basically unit of inheritance, which we are the segment of DNA that codes for a polypeptide, and that polypeptide then controls a particular trait. We know that the chromosomes they can exist as a single chromatid, or they can exist as sister chromatids attached at a centromere. In this, the DNA has already replicated to the sister chromatids will already have the same copy of the allele on both sister chromatids at the same locus. So this is what sister chromatids with DNA replicated gives an appearance to a chromosome, which you can see on this diagram at the top right corner. Then we actually have chromosomes in our body cells which exist in homologous pairs. A homologous pair of chromosomes will have one chromosome coming from the father, the other chromosome coming from the mother in case of sexual reproduction in human beings or mammals for that matter, or even plants, wherever sexual reproduction is occurring. So homologous chromosomes mean that the chromosomes are same in appearance, they are same in size, they have the same position of the centromere, and the gene locus are same as well. What do you mean by gene locus? That the location of the gene of a particular allele is also at the same location in both the pairs partners. For example, a person can be capital T, small t, small t, small t, where capital T or small t are the alleles that father or mother may have passed on at the same gene locus to the offspring. So you can be homozygous dominant or heterozygous dominant or homozygous recessive for that trait. There are some working definitions of what homologous chromosomes, genes, alleles, locus, genotype, homozygous, heterozygous, diploid, and haploid cells means. I think these working definitions are already clear to you. Sexual reproduction, when it occurs, the adult is produced having two n number of chromosomes in each cell. When the adult grows on to become, after puberty, fertile or is able to carry out sexual reproduction, the person will end up producing male gametes having n number of chromosomes, or female gamete again having n number of chromosomes, which can fuse together to form a cell of zygote, which is having 2n, or a diploid state. This is how life cycle continues. Otherwise, if this reduction division had not occurred, if the gametes were 2n, that in every generation the number of chromosomes that one would pass to the next generation would keep on doubling. So a 2n person would end up producing a 4n offspring. The 4n offspring will produce the gametes of 4n, and so on and so forth. The chromosome doubling would keep on taking place every generation, which obviously cannot support life. There's not enough room in the cells to accommodate for these many chromosomes. So to conserve the number of chromosomes, we have the process of meiosis occurring in the cells, and when meiosis occurs, the chromosomes becomes halved. So whenever meiosis is occurring, you need to keep in mind meiosis occurs in two stages: meiosis one and meiosis two. It is denoted by a Roman numeral which you add after meiosis. The stages are very similar to mitosis. Meiosis 1 includes prophase 1, metaphase 1, anaphase 1, telophase 1, followed by cytokinases. The product of cytokinases will start to undergo meiosis 2, which include prophase 2, metaphase 2, anaphase 2, and telophase 2, which again is followed by cytokinases. Meiosis 1 marks the separation of homologous pairs. Meiosis 2 marks the separation of the sister chromatids. So meiosis 1, the cell undergoing meiosis 1 will be a diploid cell, but it will end up producing two haploid cells because the homologous chromosomes have been separated. The same two cells in which the homologous chromosomes have been separated will now start to undergo further division, which is meiosis 2, and the sister chromatid will be separated once again, all over again. So the chromosomes, each cell will end up having just one sister chromatid for each chromosome. How the DNA content varies: the DNA content, half of the DNA content is passed on to each cell in meiosis 1, and quarter of that is passed on to the gametes in meiosis 2. This is how meiosis occurs. Coming to stages of meiosis, meiosis includes meiosis 1. Early prophase 1 is similar to mitosis. Middle prophase 1 allows homologous pairs to pair up together by a process called synapses, in which bivalents form. Each bivalent is actually a homologous pair. So, and the centrosomes are moving to the opposite ends of the nucleus, similar to mitosis. In late prophase 1, the nuclear envelope starts to break. The nucleolus disappears, similar to mitosis. What's new here? is the bivalents which have formed, they start to exchange their segments by the process called as crossing over. The region of chromosome involved in crossing over is called as the chiasma. So a chiasma represents a position, or even chiasmata for plural represents a position at which the homologous chromosome, they exchange their segments, the non-sister chromatids, they just overlap with one another and they exchange their parts. In this way, variation is produced. So metaphase one, they align along the bivalents line up across the metaphase plate, and they are pulled apart in anaphase one. And anaphase one, they are when they are pulled apart, since the homologous chromosomes are separating here, you can see that as the homologous chromosomes get separated, the homologous chromosomes who underwent crossing over are not homologous exactly anymore. They have exchange segment. One of the chromatid is having an exchange segment in both the chromosome which are being pulled apart for one homologous pair. This is followed by telophase 1, in which the two cells they will separate out. You can see the haploid cells have been produced, the homologous chromosomes have been separated. Similar to mitosis, nuclear envelope reform, nucleolus is reforming, cytokinesis is occurring, spindle remains, which is actually getting degenerated gradually, and the chromosomes have reached the poles, and then and you can see that the cells are undergoing cytokinesis and they are separating from one another. Soon after this, meiosis 2 begins. Meiosis 2 it begins with prophase 2. Now, all the stages of meiosis 2 are exactly similar to what happens in mitosis. So, prophase 2 marks the same nuclear envelope degenerating. Then, the metaphase 2 they start to line up along the metaphase plate. They join to the centromere through the centromere to the spindle fiber. In NFase 2, they are pulled apart. You can see in NFase 2 what's happening. How is NFase 2 different from the anaphase 1? In anaphase 1, the homologous chromosomes were being separated. In anaphase 2, it is the sister chromatids of the same being separated. In telophase 2, eventually, four cells are produced. Each cell will end up having one chromatid for each homologous pair. For instance, if you have a homologous chromosome in which the sister chromatids have been labeled as 1, 2, 3, 4 in numeral Romans, which I have mentioned here, meiosis 1 shows to you two cells forming, 1 and 2, the chromatids going in one cell, 3 and 4 going in the other cell. In meiosis 2, they all reach their destination, that is, each chromatid goes into a different gamete. For instance, if there is a homologous chromosome pair which is having the allele combination, as I mentioned here, the chromatids, the sister chromatids are 1, 2, and 3 and 4. 2 and 3 are non-sister chromatids. They form chiasma. They cross over when they cross over, and eventually when they separate out in meiosis 1, the chromatids 1, 2, 3, and 4 do not resemble one another 100%. There are new frequencies or new allele combinations which have been made here. So in 2, you can see that the last two alleles, capital G and small h, have formed here, and they have realigned themselves on this chromatid, although initially they were not present here. And the chromatid 3 is having small g and capital H at the lower part, the same part where actually capital G and small h belong. So the entire sequence of this alleles on this particular chromatid is different. The combination is different. So when the gametes form, they all go in a separate gamete. So you have 1 and 4 going in one another gamete. So these two gametes which are having 1 and 4 chromatid, they will be same as the parent chromosome, not no difference at all there. But the gamete 2 and 3, the gamete having the chromatid 2 or the chromatid 3, as mentioned in Roman numerals here, it represents a crossover chromatid going there, which means there are new allele combinations going to that gamete. So these two gametes, which are having the new variety, they are called as the new variety gametes. They are also called as recombinants. So recombinants actually are the basis of variation which comes about from meiosis. For example, if there is a nucleus of a parent cell, the diploid cell, there are two homologous pairs, capital A, small a, capital B, and small b in a particular cell. They can line up together in two ways. That capital A and small a bivalent comes in at the upper end, the capital B, small b at the lower end. For example, as shown in the diagram, on the right side, you can see that small b and capital B have rotated. So right beneath capital A, you have small b, and below small a, you have capital B chromosome. So when they separate out in meiosis 2, you can have 50% of combinations of capital A and capital B coming together, 50% having small a, small b, and again capital A, small b, and small a, capital B again being 50%. So basically, four different type of gametes can form, four different combinations can form out of these, which means capital A can come with a capital B, capital A can come with a small a can come with a capital B, and a small a can come with small b as well. This is called as independent assortment. What do you mean by independent assortment? Independent assortment basically refers to the idea that each chromosome goes independent. The genes in that go independent with respect to the genes or the chromosomes which are other than that. So a capital A going in one chromosome will not ask the capital B to come together. The capital A chromosome or that allele will go by itself. Capital B can come or small b can come irrespective whether capital A is present there or capital small a is present there. This is called as independent assortment. Lastly, this sums up how the combinations change, which I have just mentioned. So in this diagram, this is a very coherent diagram, which actually shows to you a bivalent, which means a homologous pair. You can see the red chromosome having the alleles capital A, capital B, capital C on both sister chromatids. The other partner of the homologous pair is having small a, small b, and small c on the both sister chromatids. Now, when chiasmata form, there are two chiasmas forming between non-sister chromatids, and when these chiasmata form the different parts of the homologous pair and they separate out, you can see that the four chromatid that will separate out in this way, capital A, capital B, capital C, will go in one gamete. This will be exactly the parental combination, but the remaining three are new. Small a allele have now aligned with capital B and small c. This is a new genetic combination. This is a recombinant chromatid. Capital A, small b, and small c have aligned together. Again, a new combination. Small a, small b, and capital C. Again, this is a new combination. So, genetic variation due to crossing over between non-sister chromatids and independent assortment. These are the two basic factors which we discussed in relevance to meiosis, that how meiosis is responsible for bringing about variation. The last point that you should keep in mind regarding the entire topic is that the number of chromatids is equal to the number of DNA molecules you have. For example, you were made from two cells which were gametes. So your father passed on 23 DNA molecules, it is a sperm to you, and your mother transferred 23 DNA molecules from her body to you through the egg. So when the 23 DNA molecules of your father and the 23 DNA molecules of your mother, they combine together, they form the 46 DNA molecule cell, which was a diploid cell. When those 36 DNA molecules they undergo replication, 92 DNA molecules form, but then they separate out by process of mitosis, and that's how growth occurs. In meiosis, the 92 chromosomes or the 92 chromatids, the chromosome number is always 46, but because of DNA replication, the number of chromatids they increase. So if a diploid cell is undergoing meiosis, initially it will have 92 chromatids, 92 DNA molecules in 46 chromosomes. When it splits in meiosis 1, the number of chromatids and the number of chromosomes and the number of DNA would decrease down to half and eventually to quarter. That's it.